Yeah.
On a flash drive.
It's on my computer.
Yeah, you can use it.
To you?
So, we have about 10 minutes left. We'll give you a hand signal when you're at 10 minutes, wrap it up. We can answer some questions, and then move on to our next presenters. And so, first up is the characterization of trace contaminants within lipid residue analysis of archaeological pottery. So Cadet Charles McLaughlin, tell us what you've been working on.
Good morning, y'all. My name is Cadet Charles McLaughlin, as Major Burke already said, and the title of my research presentation is The Characterization of Trace Contaminants within Lipid Residue Analysis of Archaeological Pottery. So a quick overview of the method is that we are within the field of chemical archaeology, and the questions that we seek to answer is what were the subsistence patterns of ancient people, what were they eating, but also along with that, how is their food stored, and how do they prepare it? The first step of this begins with the acquisition of artifacts. In this case, it's pottery, and so chemists or labs partner with archaeologists to obtain these artifacts. And then the next step begins with the lipid residue extraction. It involves the crushing of pottery. It is a destructive method, and then we extract the lipids or molecule of interest from the pottery fragments. And then we have residue derivatization, by which we have the lipids undergo a reaction in order to turn them into something called fatty acid methyl esters, which are a species that are chemically viable for GC-MS analysis. And the final step is the GC-MS or FID, and that just stands for gas chromatography, mass spectrometry, or flame ionization detector. These are a couple analysis methods that we use which can separate and identify individual compounds out of a complex mixture. So real quick, lipids as a target molecule. So lipids are a class of robust biomarkers which are very resistant to degradation, but they exist in nature as fats and oils. And every plant animal species has a unique way of synthesizing them using a unique combination of these fats and oils. And based on their unique makeup of fats and oils, we as chemists can interpret what people were eating. So in the table, we have a composition or breakdown of their fats and oils. And so first begins with the contamination issue. So whenever first potsherds or just sherds were analyzed, which are just low-fired pottery, which are unglazed and dark brown in color, as you can see below. These came from the Archaeological site of Green Shield in North Dakota, analyzed by previous cadets. And what we can see is that we can identify certain fatty acids, but however, there's a lot of other peaks, other bits of contamination that we just don't really know. So the GC does indicate presence of contamination. This comes from plasticizing agents and other molecules, which I'll get to later. And then further analysis showed from GC mass spec is that there was another instance of contamination whenever we were analyzing pottery. And so the main goals of this project are to characterize and identify the sources of trace contaminants present within lipid residue analysis. And the possible sources can be coming from synthetic rubber-lined caps, plastic pipette tips, and solvents or other reagents used. And so it is important to note that during the early stages of the research project, a literature review was conducted. And in one of the papers, which is very similar in nature to ours, it dealt with the lipid analysis of plants, was that contamination was noted through the use of plasticware, and the article recommended individual lab review of the plasticware before you use it. So for a quick method analysis was that the first major issue is contaminant identification, determine the identity and to what level the contaminant is present within our sample. The next is the sources of contamination. These come from vial caps, plastic pipette tips, which is an example of what we have up here, and there are two kinds used in our lab. This one up here, and then a specialized one which are used for different solvents that have different that can't exactly be drawn up with this pipette tip. And then finally, the reagents or just the chemicals we used. The procedure began with a running of controls in the reagent screening. These involve oil-free reactions as well as the independent analysis of each of our reagents, and then a baseline contaminant signature to understand what our baseline reaction data was, so we can overlay it and understand if there's any sort of contamination present. First begins with the influence of solvents and reagents. So the three main solvents and reagents used were hexane, methanol, and toluene. And as we can see with the hexane and the methanol is that there is very little to no contamination present within those two. However, with the toluene, there is contamination present. However, because it exists at the very low end of the chromatogram, right around the 5 to 10 range, it does not exist within the FAME region, which is between 20 to 35. That's whenever we expect to see our fatty acid methyl esters. So for the toluene, it is so far downfield that it will not appear or interfere with any of our FAME identification. The next is the caps. So two caps were used, a newer cap using a PTFE lining or polytetrafluoroethylene, and then a rubber lined cap. And so the PTFE caps reduce most contamination to below detectable limits, whereas with the rubber lined caps is that they present contamination within our FAME region, which exists between the 20 to 35 range, meaning that whenever we're trying to identify fatty acid methyl esters from pottery, it can lead to false positives or it can invalidate the integrity of certain our experiments. Then next we have the influence of pipette tip rinses. So the first chromatogram is an overlay of a bunch of different data, and then in the blown up region is that we have a peak that indicates octadecina-nitrile, and what this compound is used is that it is a plasticizing agent, and what it's used for is that it is a surface contaminant used in plastics. And so during the plastic making process, it helps to stabilize and to help make the plastic more flexible. And what we can see is that we would first rinse the pipette tip with one rinse of our solvent, then put it into a waste container, and then we do it 2 and 3 more times. And what we see is that the residue then goes down to below detectable limits to where it is not interfering with our data. Final step part is our conclusions. So for the method analysis, contamination is present, it is seen within control groups, and it is well above detectable limits in some cases. And then we have the reagent screening. Reagents and solvents that we use contain little to no contamination. However, any contamination that was seen within our data was not within the FAME region, not affecting the FAME identification. And then we have contaminants which can be traced back to numerous sources. These can be traced to transferable lipids on the pipettes and the caps, so the rubber lined caps using synthetic rubber lining. It can leach from the cap into our reaction vessel and into our actual reaction system. And then the pipette tips, 2 types are used, the one shown previously and another specialized one used for certain reagents. And then finally we have the method refinement. Caps have been replaced to all PTFE lines, that way we're not getting any false positives, we're not adding any extra contamination into our sample. And then the pipette tip rinsing has been implemented, which eliminate a major source of contamination, which is not only cost effective, but it's also easy instead of having to buy new caps or any new equipment. Future research, it is to apply the method of the pipette tip rinsing and the new caps to the completion of the Green Shield site. So it was a site occupied by the Archaeological tribe during the late 18th and early 19th century, and it's located on a high Pleistocene terrace above the Missouri River floodplain in North Dakota. And then that image over there is some of the potsherds, and then over here is an example of the crushing of the pottery to begin our method. Here are my sources, and I would like to acknowledge the VMI Center for Undergraduate Research and the VMI Chemistry Department for making this possible, as well as my research advisor, Lieutenant Colonel Braun, as well as the cadets that came before me to build this data up off of. And with that, are there any questions?
Okay, so I am new to this field of study.
Yes, ma'am.
Where else do you think there might be contaminants that could be picked up? So this is down at the analysis part. Is there other areas during the collection of the pottery and the processing and handling where you might see additional contamination?
Yes, ma'am. So going back to the first slide, whenever we have the chemist partnering with archaeologists, the archaeologists could not be wearing gloves. We don't really know the condition of how the archaeologists are collecting it. They could be using gloves, they could not be using gloves, and it can be found that the sweat, or if they're wearing sunscreen or some kind of oily lotion, it can leach into the pottery, which could be another possible source of contamination. And also the overusing of plastic. So if we were to use plastic test tubes instead of glass test tubes, there'd be even more leaching. So those are a couple areas of further contamination we could get. It starts from the archaeologist all the way down through certain choices of our labware.
Thank you.
Yes, ma'am, thank you.